Assalamualaikum. Hi guys. This is lecture for MLT 423 week 8 part 1. So this week I'm going to talk about complement fixation test or CFT. So what's the idea behind this CFT test? How does scientists discover that complements can be used as a principle in a test? So first we look on the nature of complement. Normally, complement components from C1 to C9 are not present in circulation in an active form. They mean they are inactive all the time. And they do not bind to free antibody or free antigen. We also need to be aware that complements are heat liable. That means they're denatured at 56 degrees when compared to antibodies which are more stable to heat. They denature at 70 degrees Celsius. This issue on stability to heat will also be going to be applied in complement fixation test. The complements to be activated when there is the formation of antigen antibody complex. So this is the classical pathway inducer. For complements to become activated, they need the formation of antigen antibody complex, as we can observe in the classical pathway of complement protein activation. So it will begin with the activation of C1, and C1 will activate C4 to C4A and C4B. C4B will bind to the surface of the microorganism or pathogens. From C4B, it will eventually lead to the formation of C4B C2B. This is also known as C3 convertase. And from C3 convertase, finally, in lytic pathway, where the pathogen will be lysed by the complements protein, formation of membrane attack complex or MAC, also known as C5b9. And then second thing that being applied for CFT is by looking on the nature of antibody itself. It is generally known that antibody have a 99% specificity and almost similar sensitivity rate. So what is actually specificity and sensitivity? Basically, specificity is the ability of the test to differentiate the negative result from the positive one, while sensitivity referring to the ability of the test to differentiate the positive result from the negative. So that means by using antibody, we can have both specificity and sensitivity. And then in CFT, we can proceed by adding an indicator system. Indicator system is the part where we can observe the result. So that's why we need indicator system. We can find out whether the specific antibody or antigen that we are looking for is in the patient blood. So by using indicator system, and the main reason of CFT is to look for specific antibody against the pathogen that we are looking for, or the pathogen or antigen itself. If it is positive, the RBC, the sheep RBC being used will stay intact. There is no hemolysis. This is positive. That means the patient do have or is positive being infected by the pathogen that we are trying to investigate. And if there is hemolysis detected, the patient do not have the infection for the pathogen that we are testing or looking for. This is how the results and interpretation in the labs. This is what you can observe by your naked eyes. Mostly CFT will be performed on microtiter plate. So as we can see here, no hemolysis means positive test. I mean the patient do have the infection, the pathogen in his or her blood. You can observe the result here. No hemolysis observed because the area surrounding the concentrated cell is clear. No hemoglobin is being released to the serum. And if hemolysis of RBC is observed, the patient is negative, do not have the infection, as we can observe here. Second, let's have a look on the CFT background. So the timeline, CFT started with in 1900 when Yushdie described the complement fixation reaction. And in 1905, the discovery of Treponema pallidum, the bacteria that cause syphilis. Syphilis is a type of sexually transmitted disease. And 1 year later, the first diagnostic test for syphilis was developed by August Paul von Weizmann. Hence the name of the test, the Bode-Weizmann test. This is a type of CFT. And then in 1946, quite an important event happened where VDRL type test was developed. VDRL stands for Venereal Disease Research Lab. And this VDRL test slowly replacing CFT for determination of syphilis infection. And in the present time now, CFT, even though are not being used for syphilis, but still being used mainly for virus and fungal infection, especially when commercial kits do not available in that particular labs. The commercial kits we are talking about here is, for example, ELISA test. Okay, let's have a look on the main purpose of complement fixation test. First, to detect specific antibody, we are talking about detecting antibody in patient, and that antibody against the pathogen that infecting the patient. And second, it also can be used to detect the pathogen itself. And CFT is being done especially on those antibodies or pathogens that do not good at agglutinating or precipitating. Thank you.